Why your DNA extraction might be failing. Ever wondered why your nucleic acid extraction doesn't yield enough DNA or RNA? Or, why your samples get degraded before you even start your analysis? Today, we'll uncover how to perform nucleic acid extraction the right way. Spekkerstoen. Stoen, ad stoen, stekkerstoen. Soen. Dapper stoen, oen neer dekker stoen, adan proen. So you can get pure, high-quality nucleic acids every time. What is nucleic acid extraction? Nucleic acid extraction is the essential first step in molecular biology. It isolates DNA or RNA from cells, tissues, or microorganisms, enabling everything from PCR amplification to next-generation sequencing. But achieving clean, intact nucleic acids depends on more than just using the right reagents. It's really all about precision and technique. How to perform nucleic acid extraction, step by step. The extraction process starts with cell lysis, breaking open membranes to release nucleic acids. Next comes protein removal, often achieved with phenol chloroform or silica-based spin columns. Then, nucleic acid precipitation, typically using ethanol or isopropanol, separates DNA or RNA from impurities. Finally, purification and rehydration restore your nucleic acids into a stable buffer, ready for downstream applications like qPCR, sequencing, or hybridization. Remember, every step matters. Temperature, pH, and contamination control all determine your yield and purity. Even a single deviation can lead to degraded RNA or contaminated DNA. Top 3 common mistakes in nucleic acid extraction. First, avoid using old reagents. Oxidized chemicals can destroy your sample integrity. Second, never vortex RNA samples too harshly. RNA is fragile and easily sheared. And third, always verify your concentration and purity using spectrophotometry. Your 2:62:80 ratio should be close to 2.0 for RNA and 1.8 for DNA. Avoid these pitfalls, and you'll save hours of troubleshooting and wasted materials. Simplify your workflow with BOC Sciences. At BOC Sciences, we understand how critical nucleic acid quality is for your research success. Our team provides comprehensive nucleic acid analysis and purification services, covering DNA, RNA, and oligonucleotide synthesis. We also supply high-purity reagents and custom synthesis solutions to ensure your workflow runs efficiently from start to finish. If you're ready to elevate your nucleic acid extraction results, visit our website, your trusted global partner in life sciences. Subscribe for more molecular biology insights, and check out BOC Sciences for expert support in nucleic acid extraction and beyond.